An empire billions of years in the making. A carnival of natural wonders. Societies of cells. Simple and complex. All connected by an astonishing web of molecular processes and global ecological systems. A web of life. These are the greatest discoveries in the history of biology. In the early 1600s, basic microscopes were in use throughout Europe. Gentlemen scientists were held spellbound by their ability to enlarge the universe of the very small. Then in 1665, an English scientist named Robert Hooke observed the tiny boxes in a sliver of cork and called them cells because they reminded him of the small rooms lived in by monks. Hooke's observation set the stage for our first great discovery. Anton van Leeuwenhoek was a Dutch merchant fascinated with science. Upon learning about Hooke's microscope, he decided to build one for himself. Dr. Gall. To find out what happened next, I paid a visit to Joseph Gall, a cell biologist at the Carnegie Institution in Baltimore, Maryland. Antoine Leeuwenhoek, what did he discover?
Well, Leeuwenhoek discovered quite a few things, but what he's most famous for are discovering protozoa, that is, small single-celled animals that live in pond water.
How did he do that?
He did it using a microscope of his own construction. Would you like to see?
Oh, yes. You have a Leeuwenhoek microscope?
Well, not an original. I have a replica of a Leeuwenhoek microscope. It consists of two brass plates with a small piece of glass, which is the lens. And the way you use this is to put the specimen on the pin here, and then you hold your eye up very close to the lens on this side, and when you do that, you can see the point of the pin, and you can put the specimen, whether it's an insect or whatever, on that pin. Would you like to take a look at it?
Yeah. It's kind of heavy because it's brass.
Well, it's brass. He made them...
In 1675, Van Leeuwenhoek was using his microscope to examine a bead of water when he observed something extraordinary. A world filled with creatures that no human had ever seen. Microorganisms. And so with this, with all due respect, primitive gizmo,
yes,
he discovered single-celled animals, things
exactly.
we take for granted
yes.
now.
What he's most famous for are discovering protozoa, bacteria, and sperm.
This thing is brilliant.
It's got a mechanical stage, it's got a focusing device, it's got all of the things that you need to look at a specimen. And it works.
Did they know what they were seeing? Do you know what I mean? Like, did they understand?
Well, I doubt that they really understood what they were seeing in the modern sense, but they were impressed that all these little things that were alive in water. I mean, people had no idea that water was teeming with organisms.
I guess I just had no idea the thing was this small and delicate, and frankly, so elegant, because it's the palm of your hand, and what you see on the tip of that pin changes the world.
It does, indeed. It was an enlargement of our understanding of the nature of the natural world, that the world out there was not just dead things, but that it was teeming with life.
Our next great discovery occurred in 1831. English naturalist Robert Brown was studying the different types of plant species he'd collected during a voyage to Australia. Brown had an exquisite eye for detail. The cells of the plants were of particular interest to him. While examining them under his microscope, he saw something intriguing. In each cell, a similar structure, circular, opaque. He called it the nucleus. Upon learning of Brown's observations, German physiologist Theodor Schwann began looking for a similar structure in the cells of tadpoles. And he found it. Each and every cell of the tadpole had a nucleus as well. It was a revolutionary breakthrough. Here was evidence that all life was connected.
In this book, Schwann describes various cell types from all sorts of organisms and identifies them by the fact that they have a nucleus in them.
How would you say the discovery of the nucleus was a great discovery?
The realization that there was a unit of structure to all organisms. This was a unit of structure not only in plants but in animals. So it united both the plant and the animal kingdoms into something that had similarities to each other.
More than a century after the discovery of the cell nucleus, it was believed there were two fundamental types of life on Earth: bacteria and everything else. Bacteria were classified as prokaryotes. These were simple, single-celled organisms with their DNA contained not within a nucleus, but by the cell wall. All other life forms were classified as eukaryotes. Their cells carried their DNA enclosed within the nucleus. But this simple classification system was in for a shock. In 1977, biologist Carl Woese was studying the genetic makeup of a methane-producing microbe when he realized it was different from any known bacterium. Its cell wall was unique. It produced unusual enzymes. And its genetic sequence was unlike anything he'd ever seen.
It became soon apparent within the space of an hour that there was something, a third thing out there.
This was the moment of discovery. Carl Woese had found a third form of life, a group of single-celled organisms that he called archaea.
We used to think there were two primary kingdoms on this planet. Now we know there are three. That was the shift. Big shift. Because all of microbiology had been structured around the idea that all bacteria are fundamentally the same. Not in their details, but in their essence, their ancestry, and their basic cell organization. Here is something that every microbiologist and biologist firmly believed in, and it wasn't true. So it does make you smile, doesn't it? Yeah. Look what I found.
What he found was a life form able to live anywhere on the planet, including the most extreme environments. Some archaea even call this home: hydrothermal vents on the ocean floor. Temperatures here fluctuate wildly within just a few inches, going from freezing to a scorching 760 degrees Fahrenheit. Archaea have also been found living miles inside the Earth, thriving in lakes of acid where even iron minerals dissolve. Today, some biologists believe that archaea are the common ancestor from which eukaryotes evolved, and that includes you and me. For Carl Woese, the discovery of archaea remains a sweet memory.
It was picked up when published by the New York Times first and the other newspapers, and then the TV came in, and I can remember walking outside of my house one night saying, when all this was happening, say, "Tonight the world belongs to you."
Our next great discovery sought to answer a question at the heart of biology: how cells form new life. You know, in the course of human events, this is all pretty recent, right?
Yes.
They found nuclei, there's cells around nuclei, animals and plants have cells.
Yes.
Then what happens next?
Well, after a gap of some 30 years or so, people began to see cells dividing.
Cell division is the process that the cell of a plant or an animal goes through to duplicate itself. While scientists had known about the process for decades, German zoologist Walter Flemming was the first to describe it and to publish his findings.
And his book was published in 1881. This book was momentous and extremely important in the history of cell biology because Flemming described cell division in a way which would be acceptable in a modern journal.
Flemming observed the process of cell division in great detail, with the help of a powerful new microscope and new dye staining techniques. With these innovations, he was able to identify structures that later were named chromosomes. And then, his great discovery. During cell division, the chromosomes undergo dynamic change. They divide into two identical parts, one for each daughter cell. Flemming called this process mitosis.
So that's the whole process of mitosis as he described it in 1881.
In a tadpole tail.
In a tadpole tail, yes.
And he knew to go to the tail because the tail is growing, or the back end of a tadpole, there's a lot going on.
Exactly, yes. He knew that if you're going to see cell division in detail, sorry for the pun.
I like the pun. Kooky for the pun.
Then that was one of the places to look.
The splitting of chromosomes during mitosis was a breakthrough discovery. It opened the door for scientists to begin understanding how one cell can turn into a complex organism made of many interacting cells. Around the time that Walter Flemming was studying the cell division process, biologists already knew that fertilization resulted from the union of two sex cells, sperm and egg. What they didn't know was why these were the only types of cells in the body capable of reproducing new life. What made them different? The first answer was provided in 1883 by Belgian zoologist Edward van Beneden. Van Beneden was studying a species of roundworm when he discovered that nearly all of its cells contained 4 chromosomes. The only exceptions were the roundworm's sex cells, the male sperm and the female egg. These cells had only 2 chromosomes. Van Beneden observed that once 2 of the sex cells came together, a full set of 4 chromosomes appeared in the fertilized egg. But just why the sex cells had only half the number of chromosomes compared to all the other cells was still a mystery. Then in 1887, German biologist August Weismann entered the picture.
He knew about the work of Van Beneden, who had shown that the sperm and the egg bring in equal numbers of chromosomes. But Van Beneden's work had not really worked out the details of how the chromosomes were reduced in number.
He saw them go in half, and he saw them recombine.
He saw that the sperm and the egg brought in half the number of chromosomes as the body cell, but he didn't really know how they got rid of the half that they got rid of. So Weismann discussed how this might happen from a theoretical standpoint. And he said, well, what you could have would be that either every one of the chromosomes would just split in half, and half would go off one way and half would go another way. Or you could have it more complicated, that the chromosomes could pair, and then instead of splitting, they just separate again.
After years of studying the behavior of chromosomes, Weissman determined that at some point a developing organism signals its sex cells to divide their number of chromosomes in half. Weissman called this very special form of cell division meiosis.
I think they will look back and see this as the golden age of biology, because, let's say, 200 years, between 1800 and 2000, we went from not knowing what distinguished a stone from a frog. Mm-hmm. Okay? And now we know the difference between a stone and a frog.
That's pretty good.
Yeah. So that, in a real sense, we've answered the old philosophical question of what is life. Maybe not to everybody's satisfaction, but we're well on the way.
Our next great discovery answered another question about the biology of life. In the late 19th century, biologists knew that during formation of the embryo, the cells of most organisms became differentiated. That is, they receive their assignments for specific jobs in the adult organism. Arms, legs, eyes. But when did this differentiation happen? German biologist Hans Driesch was determined to find the answer. In a lab experiment with sea urchins, he observed that in the early stages of an embryo's development, its cells are not yet differentiated. They can still develop into all cell types. Today, scientists call cells that can develop into all or many cell types stem cells.
Hi, Bill.
Hi. To find out more, I paid a visit to Helen Blau, who is directing stem cell research at Stanford University's Genetic Pharmacology Lab. Everybody's talking about stem cells. Cells start out, they're fertilized, then they start to divide, but they still haven't started to divide into what I would think is a liver cell, a fingernail cell, an eye cell, right?
That's right.
And what do you call that?
So at that point, they're totipotential. They have a lot of plasticity. They can give rise to all the cells of the organism. And then something changes. And we don't know what that something is, but those cells are now destined to make different kinds of tissues. And that's why we have organs such as the heart and the liver and the spleen in the adult. So then the question is, how irreversible are those decisions?
Okay.
So I wanted to challenge the idea that cells were irreversibly determined, that once a cell became liver, that was it. And so we showed that they were not irreversibly determined, and that by perturbing their environment inside the cell, you could activate genes that were usually never used. Mm-hmm. That demonstrated a certain kind of plasticity, and it's this kind of plasticity that people are trying to enlist in stem cells. So understanding the mechanisms underlying this may allow us to use the fundamental principles we've discovered in treating disease. What's recruiting these cells? What's getting them to fuse? What's getting them to reprogram so it's even more efficient? We might enlist that in what's called regenerative medicine. We're finding that cells have more potential than we thought before. So that a blood cell, stem cell, not only makes the blood, but those cells can travel elsewhere through the circulation to aid in the repair of damage in tissues such as muscle, tissues of the brain, also liver. And that's been shown in several labs now.
That's like Star Trek or something. You know what I mean? Dr. McCoy stabs the guy, and 15 seconds later, he's okay.
Well, it's not going to be quite like that.
Yeah,
We've got a long way to go. But
To gain a better understanding of the new technologies that Helen Blau is using to uncover the potential of stem cells, she showed me an experiment being conducted with genetically altered mice.
Why not? Sure.
So that is one unusual mouse.
It certainly is. It has green fluorescent protein in all of the cells of its body. Because we've been able to introduce the jellyfish or GFP gene into these mice, now their tissues glow. And you can see that particularly in the ears, the eyes, the tail.
It's spooky.
But all the tissues are green. This has enabled us then to take cells that glow and put them into another mouse and see what tissues they participate in. So in a way you can follow what stem cells can do.
It's a marker.
It's a marker.
And it just all you need is an ultraviolet light.
That's right. And it didn't exist before. And because of that, we now can follow the fates of these cells, where these cells go, what they become, in a way that wasn't previously possible.
It is fantastic. In the 1930s, German biologist Hans Krebs, the son of a Jewish physician, was forced to flee Nazi Germany. By 1937, he was doing research at the University of Cambridge in England, determined to unravel the mystery behind an essential biological process. How do the cells in our bodies convert food into energy? It's our next great discovery. To investigate the process, Hans Krebs broke open a group of cells by grinding a sample of animal tissue. He collected the liquid from the broken cells, then put the contents through a series of chemical reactions and measured the results. Slowly, a pattern began to emerge. A pattern that led to a remarkable discovery. From his measurements, Krebs determined that sugar molecules from digested food go through a cycle of various chemical reactions inside the cell. This cycle results in the production of the energy-rich molecule ATP. This molecule provides us with the energy we need to make us go. And the cycle of chemical reactions is known as the Krebs cycle. It was a milestone discovery in the development of biochemistry. The Krebs cycle opened the door to a deeper understanding of how cells function in the human body. Just like our next great discovery. In the mid-19th century, scientists using powerful new microscopes found something never seen before, a mysterious structure lurking inside nearly all types of cells. It was equipped with two membranes and had the ability to change shape. Over the next 100 years, several biologists combined to uncover the secrets of this biological wonder. They called these structures mitochondria. Among the scientists involved in the first wave of discoveries about mitochondria was Britton Chance. His contributions included the invention of a dual wavelength spectrophotometer, a device that provided scientists with a clearer picture of how mitochondria function. Today, he's a professor of biophysics at the University of Pennsylvania.
I made a gadget. This gadget would look through the turbidity of these organelles because they had membranes and they had cell walls and they had things which scattered light, and you hold them up to the light, all you could see was a fuzz. Well, we made an instrument which got rid of the fuzz and found two components that linked the mitochondria to Hans Krebs' citric acid cycle. So this sort of tied the whole thing up and made links between enzyme action and the whole energy chain. So it sort of glued it together.
With the help of Chance's invention, scientists found that the mitochondrion provides the energy that enables a cell to work and the body to function.
The mitochondrion has to be a very efficient way of using the foodstuff that the host cell gathers. And this is called a phenomenon of respiratory control. To the layman, it means that he can exercise like hell when he wants to, and all he has to do is stop to recoup. In other words, he doesn't have to take a pill, he doesn't have to take an injection. His mitochondria will reconstitute the energy deficit. Having done that, they'll go to sleep. So I think especially in these days of Olympic sports where everybody's pushing their ATP to the very limit, it has a real significance.
Britton Chance appreciates that significance as a scientist and as an athlete. In 1952, he won a gold medal as a member of the U.S. Olympic sailing team.
I think discovering the components, studying mitochondria in vivo, finding out how they work, showing how important respiratory control was and how it worked, and of course studying free radical generation, which now is recognized as not so dangerous but a signaling process. And that work sort of keeps me off the street now, but I'm working more on brain and cancer because of the implications of mitochondriology there.
At the end of the 19th century, one of the great puzzles challenging scientists was how the billions of nerve cells that make up the human nervous system communicate with each other. They knew that each nerve cell, called a neuron, carries electrical impulses, and many believed that the connection that allows the impulses to jump from one neuron to another was electrical as well, until our next great discovery. In the early 1900s, British biologist Henry Dale was conducting a series of lab experiments studying the physiology of nerve impulses. In one of his experiments, he injected adrenaline into a cat, expecting the animal's heart rate to increase, but nothing happened. Suddenly, Dale realized his mistake. It was the same cat that he'd already given another drug to, one that slowed the heart rate. With this mix-up, Dale realized he was onto something. If a drug could interrupt the nerve impulses that govern the beating of the heart, then the connection between neurons must be chemical, not electrical. Dale turned to his colleague, German biologist Otto Lowy, for help. To test Dale's hypothesis, Lowy conducted an experiment using two frogs. Lowy put the first frog in a saline solution. Then he electrically stimulated its vagus nerve, the nerve that controlled its heart rate. The frog's heartbeat slowed. Next, Lowy took some of the solution and applied it to the heart of the second frog. What happened next was extraordinary. Without any electrical stimulation of its vagus nerve, the second heart slowed as well. This was the moment of discovery. Lowy realized that the vagus nerve of the first frog had released a chemical which had directed the frog's heart muscles to slow their contractions. Here was proof that the transmission of impulses among nerve cells and then to the heart muscle was chemical. Lowy had discovered the existence of the first known nerve chemical, now called a neurotransmitter. Today we know there are many neurotransmitters in the human body, and researchers are using that knowledge to learn more about how the brain works and the chemical messages it sends. For example, low levels of serotonin have been connected with depression, alcoholism, and anxiety disorders. But as our next great discovery revealed, the nervous system isn't the human body's only method of communication. In 1903, two British physiologists, William Bayliss and Ernest Starling, were investigating the food digestion process. From their studies, they determined that the body produced digestive juices that helped break down food once it moved from the stomach to the intestine. But what caused the digestive juices to flow? To find out, Bayliss and Starling conducted an experiment. They took a blood sample from a dog that had just eaten a meal. They injected the blood into a second dog, this one with an empty stomach. Then something astonishing happened. Without any food in its belly, the second dog began secreting digestive juices from an organ called the pancreas. How had this happened? After more testing, Bayliss and Starling finally had their answer. As food reaches the small intestine, the intestine produces a chemical substance which is then carried through the bloodstream to the pancreas. Here, it stimulates the pancreas to produce secretions that help digest the food. Bayliss and Starling called this chemical secretin. The first of what they realized was a never-before-seen group of chemical substances. They called these substances hormones. Since Bayliss and Starling's discovery, more than 50 hormones have been identified. Hormones that are produced by glands and tissues and carried in the bloodstream as the human body needs them. There are hormones which affect the body's growth, metabolism, heart rate, and blood sugar. They even help us prepare for reproduction. Estrogen, for example, is one of several female hormones that helps prepare the womb for the baby and the breasts for feeding. So far, we've seen discoveries that revealed the biology of life on the microscopic level. Our next great discoveries opened our eyes to the biology of the macro, those systems on which all life on the planet depends. The year is 1771. Chemist Joseph Priestley conducts a series of experiments to see what happens when he heats various substances in closed jars and collects any gases coming off. In one of the experiments, he put a lit candle under the jar, but the flame quickly burned out. In Priestley's words, the air inside was injured. Curious what effect this injured air might have on a living thing, he placed a mint leaf inside the jar and left it there for 10 days. When Priestley checked back, incredibly, the leaf was still green. Somehow the air inside the jar was fresh again. Priestley speculated that the mint leaf must be responsible for this transformation. But how? This question remained unanswered until a Dutch scientist named Jan Ingenhousz decided to duplicate Priestley's experiments. Using plants, Ingenhousz witnessed the same beguiling phenomenon. After conducting a series of more tests, Ingenhousz began to realize what was happening. The ability of a plant to transform the air inside the jar only worked if the plant was exposed to sunlight. This was the breakthrough. Together, Priestley and Ingenhousz had discovered photosynthesis, the biochemical process by which plants trap the energy of sunlight and convert it into chemical energy. Photosynthesis removes carbon dioxide from the atmosphere and replenishes it with the gas on which our survival depends, oxygen. The same gas that Joseph Priestley would discover 3 years later in 1774. Our next great discovery has no discoverer in the classic sense. Many took a hand in expressing and shaping the idea over several centuries. It's the discovery of tropical biodiversity. And today, it's fundamental to our understanding of the biological world and our dependence on it. The term biodiversity refers to the variety of living things on Earth and their interactions with the environment. The tropical rainforests are the richest resource of biodiversity on the planet. In the 15th century, Italian explorer Amerigo Vespucci visited the tropics and wrote in his journal, "What should I tell of the multitude of wild animals? So many species could not have entered Noah's Ark." Today, we continue to be awed by the biodiversity of the tropics. An estimated 50% of all the species in the world inhabit the tropical rainforests, an area that occupies only about 2% of the land on the Earth's surface. The Amazonian rainforest in Brazil has nearly 500 species of trees growing in an area of just 2.5 square miles. Compare that to the total area of the United States and Canada, which combined have only about 700 species of trees. But just why is the discovery of tropical biodiversity considered so important? Because not only did it enable scientists to better understand the natural systems at work here, but it helped them begin to harvest an extraordinary array of resources which touch our lives every day. From the production of more food and better industrial products to the development of new medicines. For example, many of the most promising anti-cancer drugs come from plants found in the world's tropical rainforests. But those benefits have come at a steep price. Scientists estimate that the loss of tropical biodiversity is happening at an alarming rate. Millions of acres of habitats have been wiped out. Countless species have gone extinct. It's estimated that with the current rate of deforestation, all tropical rainforests could disappear by the year 2030. That disappearing act has far-reaching environmental implications. Because, as our next great discovery revealed, all the Earth's biological systems and the communities of life that depend on them are connected. The Dust Bowl of the 1930s. After years of drought, the once fertile American Midwest became a wasteland. Without rain, farmers continued to plow and plant, hoping that the nutrients in the fields would somehow be replenished. But their efforts only made things worse. The topsoil turned to dust, and the wind took its vengeance. For British ecologist Arthur Tansley, the Dust Bowl was proof that scientists, and the public at large, were too narrow in their view of the physical and biological world. They did not understand the dynamics of the environment as a whole, a perspective that helped explain the cause and effect of events like the Dust Bowl. He advocated a new concept, what he called the ecosystem.
The word has a simplicity to it. It's understandable by the ordinary person. It can be applied effectively, but it also has a scientific significance in that it brings together ideas from a variety of sciences. And
to understand the impact of Tansley's idea, I paid a visit to ecologist Frank Golly at the State Botanical Garden in Athens, Georgia. Why was this a revolutionary shift in thinking?
At that time, ecological work was strongly involved with the description of nature.
In the systems approach, you are actually going further. You're experimenting. You're looking for patterns that are persistent across a variety of locations and species, and then you're making statements about the way they perform that can be tested. This moves ecology into the same venue as physics and chemistry and molecular biology.
And so Tansley took that word system
and applied it to ecological studies. He tried to convey the idea that ecology is part of a hierarchy of physical systems from the universe at one extreme to the atom at the other.
So somewhere in the middle would be solar system.
Yes.
And then down below that would be
ecosystems. Yeah. Back here is something I want to show you. The quantity of silt that you see in the river right now is due to the erosion of the farmland that's upstream. This is a opportunity to see what we could say was an ecosystem defined physically, Bill, right here in front of us.
It has boundaries.
It has boundaries. It's formed through a manipulation of the flood cycle, the river.
So this is a floodplain.
That's right. It receives water from the Oconee River. Periodically we get enormous rainfall and the river floods and would then send water into this sort of a area. We might be standing chest deep in water during a flood. Presumably at one time it was a forest. We see the remnants of the trees. But because of flooding and the persistence of flooding, the trees' roots were unable to respire and the trees died. You can see if you look around yourself here how beautiful the recovering forest is in its shape. We've studied that in great detail using an ecosystem approach. So that now we're able to talk about how to rebuild ecological systems here in this region by manipulating the various species and the environment to speed up the process, to get the endpoints we want to emphasize, and to do it at lesser costs. And that's a very positive step because we can rebuild nature to meet certain human needs, and so that it functions and maintains itself on its own under solar power.
Solar power, yeah.
Under solar power, which is driving all the cycles and everything else.
Our journey through the greatest discoveries in the history of biology has revealed an amazing diversity of life on Earth. And it's given us insight into how that life works, from the life cycle of the smallest cell to the systems that govern the physical environment of our entire planet. But if there's one insight that resonates above all others, it's this: the search for more discoveries about life is never ending. That journey goes on forever.